As we started this project, I wanted to try and use something very unique to represent the cycle of photosynthesis. Hopefully this depiction of photosynthesis gives you a feel for what the process looks like as a whole in a fun and new way. Let me start by telling you what we are looking at. The board as a whole makes up a section of the chloroplast where this reaction would take place. The first half of the board represents the light reaction, which would take place in the thylakoid membrane and use molecules from the thylakoid space, which is inside of the phospholipid bilayer, the thylakoid membrane. In an actual chloroplast, there would be tons of thylakoids stacked on top of each other, making a granum, but we're going to focus on one specifically. The second half of the board represents the stroma, the part outside of the thylakoid membrane, but inside the chloroplast. Okay. Now that we know what we are looking at, I'm going to go into a little more detail about the different pieces of the game and what they represent. We're going to start, ironically, at the stop sign. This piece represents photons that are stuck in one of the light harvesting complexes of the chloroplast. Right next to the stop sign, you'll notice a little blue mouse. This guy represents water that enters the system through the xylem and phloem that reach the leaves from the roots. The photon causes a reaction in the complex that causes the water molecule to split and become reduced, thus giving off an electron that is passed to the primary electron receptor. This pass is represented by the boot, which becomes excited when the stop sign hits it. The bucket on the stairs represents the primary electron acceptor, and the marble inside is the electron that we just released from the water molecule. We followed this electron through quite a bit of stuff, so don't lose track of it. A side note is the leftover piece of the water get used as well. The hydrogen ion is released into the thylakoid space to be used later to make ATP, while the oxygen atom binds to another oxygen atom and is released into the air as oxygen gas through the stomata. The light reaction process occurs in both photosystems 2, also known as P680, and photosystem 1, also known as P700. Photosystem 2 is represented by the blue stairs, and photosystem 1 is represented by the red bathtub. And together we can show the linear electron flow that takes place during the light reaction. Once the electron is accepted by the primary electron acceptor, yellow bucket number 1, in photosystem 2, it's put on the first section of the electron transport chain, which is an electron carrier PQ. This is represented by the red rain gutter. When the electron reaches the cytochrome complex, it causes a reaction that allows hydrogen to enter the thylakoid space from the stroma. This is represented by the marble leaving the rain gutter, aka PQ, and going into the yellow bucket number 2, also known as the cytochrome complex. That action releases the wrench or hydrogen ion and brings the marble to the short ramp, also known as PC. A quick side note is this causes a proton hydrogen plus gradient inside the thylakoid space, which is important to the process of photosynthesis. To continue, the electron travels down the second half of the electron transport chain, which is the electron carrier PC, or the red short ramp. Now we have entered photosystem 1, and the process is similar to that of photosystem 2. The electron is passed to a primary electron acceptor and put on an electric transport chain. However, this time, the electron transport chain is made up of FD, which is represented by the hole in the bathtub. Then it is put into NADP plus reductase, represented by the green mouse, which oxidizes NADP plus and a hydrogen ion to create NADPH, represented by the left elbow of the diver. While the production of NADPH is taking place, the hydrogen ion, or the red wrench, that has traveled from the stroma to the thylakoid space by way of the cytochrome complex, has made its way to the ATP synthase structure, represented by the plumbing, and is now represented by the red mouse. The hydrogen ion passes through the ATP synthase, where ADP and a phosphorus molecule are joined together to make ATP, which is represented by the right elbow of the diver. Once these two molecules are created, they travel to the Calvin cycle that takes place in the stroma. This switch from the light reaction to the Calvin cycle is represented by the diving board when the diver jumps off the diving board. When the diver hits the wash tub, this causes the cage to begin to fall. The falling of the cage on the cage pole represents kind of a lot, so stick with me. The initial quick drop of the cage represents the short-lived carbon fixation of rubisco. Then it continues to drop, representing the split of rubisco into 3-phosphoglycerate. Then ATP from the diver's elbow adds a phosphorus molecule to the phosphoglycerate, making 1,3-biphosphoglycerate. As the trap settles onto the ground, this shows NADPH joining to the 1,3-biphosphoglycerate, making G3P. This is represented by the yellow mouse in the trap. One of these molecules goes on to make glucose and other organic compounds. This is represented by the single piece of cheese. The other 5, represented by the other 5 pieces of cheese, are used to regenerate rubisco by joining with ATP and creating RUBP. This returns to the beginning of the Calvin cycle to repeat the process. And that completes the light reaction of the Calvin cycle of photosynthesis. I hope you've learned a lot and the process helped walk through photosynthesis in a fun and new way. Now let's watch it happen all together.